Just the reptile corner of the brain.
Is that one there? No. Yeah? No.
Yeah, that's a pronoid.
There we go.
Yeah. I don't see any predators though. I see some squat lobsters.
Right.
Yeah. Sea stars tend to be the easiest because we have a reasonably sized reference data set and other similar data for other geographic regions. And most of that is sea stars on primnoids.
I'm screwing up here. I'm gonna let you get deeper than me. What am I thinking? I should be on the other side of you. That way you can actually see me. So I will, I'll lateral along the cliff face for a while. So you come past me.
Copy that.
Sorry.
So we know what's going to be the most common thing we're going to see. What do you think is going to be the most uncommon thing we're going to see? Or most rare thing that we're hoping for?
That sounds like a warm-up question.
We're looking for one of those sea stars with its insides out on a coral.
I would be really excited to see that.
Going to town. In such a fashion where we can get some really cool, gruesome video of it.
It's like slow motion horror movie.
It is a slow motion horror movie. I think that's what Steve's looking for.
So Matt, do you know how sea stars eat?
With their beaks?
That's a good guess, but they don't have beaks. It's pretty cool. So they have a giant stomach inside their body, and sea star's mouth is on the bottom of their body. Like you could imagine taking one and flipping it over. And they take their stomach and they actually take it outside their body. They evert it, is the term that we use. And it goes out of their mouth and it wraps around whatever they want to eat. And then they secrete like digestive juices with their stomach outside their body, and it's pretty nimble. Like it'll wrap around different branches and, just kind of like envelop a chunk of a coral. And they digest it and they suck all the good bits back inside their body.
That's really cool. I didn't want to sleep tonight anyway. Yeah, that's... I didn't know that. That's interesting.
I almost got uninvited from a Thanksgiving from discussing that with my little cousins. They thought it was really cool and really not dinner table appropriate. So yeah, that's a cool one to see.
Do all sea stars have that sort of thing, or is that just one specific type?
I'm ready.
Steve? I think all sea stars eat that way.
Yes, except the brachyncids.
Okay.
Probably don't do that.
They kind of pass the food down to their mouth?
Yep.
Okay.
The slime stars, the pterastrids, probably don't do that. They're more of deposit feeders.
Fantastic.
Probably others. Not my specialty.
Help us, Christopher Ma.
So do sea stars, do they hunt, or do they just kind of float around and then grab onto stuff?
Can we take a look at twig in the back right here? White twig.
Sure. No.
I think they hunt. Maybe the detritivores that we just mentioned, the slime stars, that would just be moving around kind of like a sea cucumber, picking up stuff that's sunk to the seafloor, but I don't know that we have great answers on how sea stars find the particular coral they want to eat, but once they're there, they can just climb up the branch and keep eating for a really long time.
Zoom in on that one for me, Dan.
They have chemosensory structures that would allow them to move. It's kind of like a, chemosensory Doppler. They have on their arm tips, they have chemosensory receptors, and they can sense concentration gradients across their bodies from arm to arm.
This is the lake warmer.
Did you get an ID on that guy, Steve?
No, not quite. It's unique enough that it's worth setting down. There's another one up here if that's easier to get at.
Where'd it go?
Yep.
Oh, I see it.
Yep, that one or even this one. All three of these are the same thing.
Should be able to, I'm hitting something. Try this one up here. Okay, try to zoom on that one, Dan.
Sure.
Get a little better shot of it. Hold on, I'll get a little closer and get the ROV
Cool.
stable.
All right. Well, that kind of helps. That's this tells me that it's likely an acanthogorgid, so a different family from the yellow fans we've been seeing. It looks like the polyps are retracted or semi-retracted, and yeah, you can see sclerites in the body wall, and sclerites sticking out through the tentacles. So these are, it's we've seen a couple of colonies so far that are unbranched, but it's most likely that they're very small. On the other rock adjacent to us, there's a branched colony, so I'm pretty sure that these are probably just very small or young colonies that have not yet branched. But it's good to see that. All right, all set there.
You want to look at the branched one, do you?
No, we don't have to. No, I imagine we'll probably see more. Immediately up and to the right here, there was something very, very faint I wanted to check out.
That was a great shot, Dan and Dan. That was so cool
Right
watching the
here.
snow drift by.
Okay.
I see nothing there. I see a rock.
Right there.
Oh, look at the star.
It's very, very faint.
Oh, now I
pink.
see it. How do you see that?
You can snap in.
It's almost
amazing.
invisible. Look at that.
Okay, Dan. Do a little zoom there.
So to answer that question in the chat from Jason, how long is this dive scheduled for?
Okay,
We are
you'll go a
scheduled
little
to
tighter
lift up
there.
our ROVs around midnight our time, and we start around 4 a.m. our time. So 4 a.m. Hawaiian Standard to midnight Hawaiian Standard.
Well, it's a black coral. It's very unusual, probably a schizopathid, the same family that bathypathes and others belong to. It's kind of easy to see the pattern of the branches here, but I think just a good shot would be enough.
It looks like some of the top is missing. Would I be right in saying that?
No, it's probably just small colony. So there may be some branches here, like there's a gap that fell, but that looks kind of a normal top for this. I would hazard bathypathes would be my guess, but I'm not sure. It could be alternatipathes as well. Okay, all set.
Roger.
Thank
Moving
you.
on.
Looked smaller and more finer than some of the other colonies that we've seen so far on the dive.
Yeah. Roger. Let me, how deep are we? Where are we? 1,600? Got a little
Here's
closer.
another interesting question for the chat for you, Steve. I see a lot of scars on the rocks from previous attachment. Is there any way to tell how long ago life forms used to be alive on that exact spot?
Maybe.
Yeah, let's go for it.
I imagine so. It's calcium carbonate or some mineral of calcium carbonate. Some corals produce a arachnidic basis, some calcitic bases. But I imagine you could possibly date that material.
Coral and a sponge.
If you could get it off, I imagine it would be very difficult to get off intact
This is
using the
awesome.
tools that we have. So potentially dateable, yeah.
How do you date sponges and other things that we find off of the bottom of the sea?
You sink a ship, and then you come back every year and see how big they get.
You know, I kind of like that method.
Serious.
It is a method.
That's
That
a
is a
legit
method.
method.
Yeah.
The other
Okay.
thing is you lay a cable, a big fat cable, or a pipeline, and they start growing on that. It's got to be a deep water one, though.
These are all Iridogorgia bella here.
Beautiful.
We've seen there's two species of overlapping depth range right in this spot, Iridogorgia bella and Magnispiralis.
Magnispiralis is the big spread out one though, right?
Yeah, very, very wide coils.
Wide
Bella
coils.
has very narrow coils.
And that sponge to the bottom left, I haven't seen something like that. Is that just a different variant of glass sponge? Like, What exactly is that?
Yeah, it's likely a glass sponge, probably in the family Pheronematidae, and possibly Poliopogon, different species than we were seeing mostly yesterday. But Poliopogon is kind of a catch-all
Come down.
That is coming
group
tight.
that has a lot of different morphologies, so probably needs some taxonomic work. So it might be a different genus after
Come down a
after
couple
everything
meters
is
there.
Coming down.
resolved.
Look at this
Okay.
bamboo.
But
That's so
consistent with
cool.
That is-
Poliopogon so far.
A giant piece of bamboo. That's huge.
Okay, so if you're watching on, not watching on channel 2, click over there for a second. You have a really cool view of Hercules looking down at this, and you can, get a little bit of the scale that we are not on the seafloor. We are a pretty significant
As
ways
close
up.
as I can get with the current tether we have.
That's all right. Let's turn around, or well,
Keep
however
moving?
we get there. Yeah, go head towards 4.
Yep.
Because I want to try and keep on schedule, and get to the end of the dive with some time to spare.
Roger. I come back a little closer to you.
The hardest thing
Wanna
to
come
do
up
as
a
a
little
watch
bit?
lead is when to say enough is
I'm gonna have a
enough.
good view because I'm coming down backwards here. So
Gotcha.
There's a lot of cool stuff to choose from down here. So what do you try to prioritize?
Well, it's a balance of things. You don't know what you're going to. So there's a famous maybe famous quote, famous movie line from Life Aquatic that says,
such a great
on exploration, they're talking about exploration. We don't know what's going to happen. We just go down there and we film it.
At the
That's
moment it does.
that's the
'cause
idea.
I'm way out there. Yeah,
And that's
but
kind
I'm
of
gonna
like
come
what
closer
we're
here.
doing
I'm
here.
just
We don't know
going to come
exactly what we're
closer
going to
along the
find, but we're
overhang.
trying to plan a dive that covers a range of objectives, from looking at and inspecting, community diversity, sampling rocks, trying to finish the dive track as much as possible, because the dive track also tells us something about spatial distributions across the seamount landscape. So where do we find the most corals? Where do we find fewer corals? And it's really all important because, just having a dive worth of presence or absence data, plus a good multi-beam map.
moving if
Oh
you
yeah,
want.
there's a.
Yeah, I'm good.
If there's no live tissue in there, it's not.
Is
It's
that
not going to
Magnus
be a
Borealis?
collection. Yeah, that's Magnus Borealis. It's that way.
I'm not on the kukui level, but I'm doing like one thing at a time.
Oh no, that was a good one. I need to get on your level.
Teamwork.
Makes the dream work.
So with this, we can start to, if we have a ridge or a seamount covered, on at least one side, but preferably multiple sides, we can start to do something called predictive habitat mapping or predictive distribution mapping, where we could use all the different features like slopes and depth, some oceanographic variables, and start to determine probability maps for where we might find certain species based on
You hold your
ground
depth
truth
there.
records
In
that we're
theory,
seeing here with
you're
our
gonna
eyes.
you're going to get deeper, so I'm going to come
And sometimes
towards
that
you.
can be really
Roger that.
useful
In theory, you're going to get more altitude as she moves down the hill there.
because it allows us to
And
apply
I'm going to come towards you, so
probabilities to where we might
more
find
uphill
certain
from me.
species of interest
Okay.
over very large areas without having to physically be there.
These long bamboo whips really look like lines on the seafloor.
Yeah,
Amazing.
Is that the skeleton of a bamboo coral?
that's what it looks like. Yeah.
Oh wow.
So can
There are a couple.
bamboo coral, like the skeletons, can they bleach like anything else? Or do they have like a sort of different makeup than a skeleton like me or you?
I think I'm not sure about the mineralogy if it's different from human bone, but it's similar. It's calcium carbonate, but at this depth, it's not going to bleach. More than likely, what's going to happen is it's going to dissolve or erode. Mostly dissolve. Like if you look at this one here, you can see how some of the internodes, the pieces of calcium carbonate between the black bands, are a little emaciated and skinnier than the node. That's not how it usually is in life. Usually the calcium carbonate node is sometimes thicker than the proteinaceous node, but the difference is that calcium carbonate is more soluble at these depths, so it's going to actively dissolve into the seawater, whereas the protein is not. So that's why the nodes look look like they're bulging out from the skeleton.
So that coral was on its way out?
Most likely, yeah, if it wasn't covered with polyps, it was dead already.
So I know that black coral was used for jewelry, and other sorts of coral are used like in ingredients and for making commercial items. Is bamboo coral, are they harvested for anything?
I don't believe so, but I don't have a complete knowledge of that. It's worth saying too that coralliids, precious corals, and black corals are protected under international conventions called CITES. So you can't, for example, buy and take across international boundaries these protected species like black corals and precious corals. The Convention for the International Trade of Endangered Species. Those are two groups that are protected.
Oh wow, look at this rock.
That's a nice spot.
It's beautiful.
This is
Good
a
diversity here.
Wow,
Maybe
look at that.
15 species, 10 or 15 species.
Nice.
Pretty much everything we've seen so far since we've got up into the top of the summit.
It's our little
Put it all
collection.
together.
It's a nice little summary rock.
Yeah, great, exactly.
For folks just joining us on the spot, check out all the species that we have seen thus far.
Can you name all of the species? Put
Look
in
at
your
the
answers
sponge.
now.
Oh my gosh.
Goodness.
Wow.
Love it.
That's a big boy. Is huge. Huge.
Did I say Magnus borealis there too?
Yeah, I think so.
Yeah.
Wow. So Steve, is it possible, for example, if someone were to introduce a new type of coral to an environment, could that coral become invasive or would it just die off?
With that question, to the best of my knowledge, I don't think there's ever been an invasive deep water coral species, only because ranges at this depth are sometimes several thousands of kilometers. So, trying to understand what is invasive in a certain area is difficult because things have very wide ranges and sometimes we don't even know their absolute range. But to also understand that, you're talking about decadal timescales because things in the deep sea down here, remember we're 1.7 degrees C, something like that. It's very cold.
Yeah.
Processes happen very, very slowly.
I think it's an interesting question. Some people talk about, remediation or kind of like replanting corals as a way to restore areas. And there just aren't successful examples in the deep sea of being able to kind of like repair damage once it's done. It would take, and even to test that theory would be such a wild undertaking, right? You'd have to commit to like 100 years of study, returning to the same sites, seeing if there's growth, seeing if there's success.
I can think of someone who's done that, or one group of people that's done that.
That have replanted?
No, not necessarily replanted, but like monitored.
What group of people?
Give me a second. Let me get a little closer.
Wham
Just
stick.
stretched out here.
The Whiffle assisted measure.
Overhanging cliffs are really cool.
Yeah.
From the Gulf of Mexico.
Whiffle bottles, yeah.
Back in
Yes.
2014 and 15, we looked at recovery of some species in the Gulf of Mexico that had been impacted by oil and dispersant chemicals from the Deepwater Horizon. We went back there a couple years in a row at least, but there was a longer time series, and they evaluated whether there could be recovery, to what degree some corals recover that were impacted. And they were imaged successively year after year, sometimes multiple times per year. And in order to get good imagery, you had to use a wiffle ball and a stick to get a good scale.
You mean a non-wiffle brand?
Yeah. Oh.
A non-wiffle brand generic ball with holes in it?
Right.
We got a cease and desist from the Wiffle Corporation during that expedition. But they sent us wiffle balls, authentic regulation wiffle balls and bats. So they're a really cool company. They just were protecting their trademark.
Wiffle, if you want to sponsor the EV Nautilus, please go ahead.
We'd love to continue the partnership of 2015.
With Wiffle.
Wiffle of science.
So, Steve, when you collect these sponges, when you collect these corals, are they usually pretty heavy? Like, how much would they weigh normally?
They're very light. Usually, it depends on the group, but sometimes, tens to hundreds of grams, maybe.
It's a good question. What's their BMI? Probably less than mine.
Yeah.
I feel like that's more of like a sea cucumber measure. What's the BMI of a sea cucumber?
What is the BMI of a sea cucumber?
Very good
Liz,
question.
we have questions.
I think Liz is probably asleep.
Wake up, we have questions.
Screw you and your sleep schedule. We need to know about sea cucumbers, damn it.
So if you want to check out Liz's profile and learn a little bit about their science,
Sure.
check out
Neat
the team
little
page,
species here.
and you can click on the bios of everybody on board.
So this is actually
cool.
a new species
Cancel that
undergoing description currently.
Oh, wow.
Cancel that move.
This has been collected by this ship as well as I believe others, but this is a coral that is called Rhodaniridogorgia new species. It is a very highly branched Rhodaniridogorgia. So if you see, it has some characteristics of Iridogorgia that we saw earlier, the coils, but instead of having very helical coils, it's more of a wavy axis. You can see that really clearly on the left-hand branch. But this was sampled last year up in a few places, I think, but most recently during our Voyager seamounts.
Oh, cool.
I believe expedition in November last year.
That's exciting. Who's working on describing it?
That's a good question.
Okay.
I don't know who's actively working on it right now, but I know that from talking with colleagues that it is of interest, and these things take time sometimes.
Does Rhodaniridogorgia also have like a really tall kind of single plume version?
Yes. Yeah. The
Expression?
There's a species called Rhodaniridogorgia superba. Yeah, you'll see that. We're a little deep right now.
Okay.
But you will see that as you get usually shallower than about 1,000 meters. It is a single, stalk, single coil, much like Iridogorgia. But I've seen this enough now to know that not to get too excited, even though it's a really nice looking colony.
Why can't we get too excited? It's amazing.
Excited enough to want to sample it again.
Okay.
I think we've got plenty of material. We got a good sized piece last year.
Solid.
But there's lots of associates in here.
There really are. So you can see crinoids. Some of those things that look like crabs are squat lobster. They have those really extended long pinchers. You see, I don't know, you think about them being well adapted. The one in the back, you can see with the pinchers up, trying to catch marine snow or plankton.
You'll notice.
There's one.
Oh, there it goes.
Zoom in a bit. Half zoom for me, too.
What's really cool about these corals is that I've acquired a new term that I really like for corals that have lots of associates with them, and that is the biodiversity multiplier. Because you find a lot of species here that you don't typically find anywhere else. So you wouldn't see these squat lobsters on any other colonies necessarily, but in one spot you can see 10 species within one. So they really do add to the diversity of an area, much like a coral reef does, but In this case, it's just a single coral head, coral colony.
So when we see all these brittle stars and all these squat lobsters, is it possible that we see any bigger fish living in these kind of areas? Or do we know if we've seen any bigger fish living in these areas?
It's possible. Haven't seen them. A lot of bigger fishes don't typically come around Hercules very much. Typically stay their distance, but it's possible. You know, some fishes may lay eggs, for example, like things like cat sharks. We're a little deep for them as well, but they've been known to lay eggs in chrysogorgid colonies like these. All right, I think we're all set here. Keep going.
I love that term biodiversity multiplier. It really, underscores why you would want to protect areas like this within Pacific Remote Islands Marine National Monument.
Can we drop a target there and call this like Coral Peak?
Coral Clubhouse.
Coral Peak, I like it.
We're not a creative lab, but we know what we're doing.
Speak for yourself about knowing what you're doing.
It's my one day my skill, my dream skill to be able to come up with good, witty acronyms. I feel like that's what you need to succeed in life.
Witty acronyms.
Yeah.
Nice.
Superpower.
I
So
would
Steve,
settle for
I see sometimes some shrimp will either fly by Atlanta or bump into Hercules. So are these shrimp, are this like the same kind that we as humans would eat, or is it an entirely different species?
Probably wholly different group, yeah. Most of what you're going to see here are nematocarcinids or Penaeid shrimp. I don't think any of those are commercially fished, but I'm not certain on that. You might see small mysids, though. Mysids can be found in shallow water, but they're never commercially fished. They're very, very tiny.
So with all these brittle stars and all these like kind of sort of squat lobsters living in this coral, why do the brittle stars wrap their way around coral? Someone in the chat wants to know.
It's a great way to hang on, and especially if you're in an environment where there are strong currents. There's a Narella macrocalyx there. It's a very distinctive branching pattern. Yeah, they're mostly planktivorous, so they're probably extending an arm or two out into the flow and grabbing particulate matter, food from the water column. It's more advantageous for them to be higher up in the flow because it gives them access to a faster supply of food per unit time. So it's a very energetically favorable place and activity to be engaged in.
And another question from the chat. What makes certain coral hard and others look more soft and flowery?
It's in part due to their taxonomy. So, a lot of hard stony corals are have lake characteristics. They share hard stony arachnitic skeletons, whereas the octocorals like these and black corals will often have a harder skeleton but softer tissue, and they're branched in such a way that it helps them feed more effectively. Stopping and slowing down water, so it can capture particles out of the flow. Reefs and stony corals do a similar thing, but they produce a much harder skeleton that acts in a similar way, slowing down water.
Okay, Lynette, I'm good for a 20. Yep.
If you're talking about shallow water environments, though, you're also talking about space competition, access to light, which are needed to calcify.
Seeing a nice low bait flow here on the rock.
So it's a nice low bait flow from a bad low bait flow.
Oh, that's just an adjective I'm adding. It's not, just looks good, I guess.
Fair enough.
You can see the lobes
nice
well defined, I suppose.
Follow beans.
It's not a scientific good.
Really cool
nice.
old sponge skeleton.
Large old glass sponges, possibly farades.
And was that gold coral or golden coral that we just passed?
Can we look at the purple coral down in the middle?
Sure.
Just want to do a double check and see if that's Victorgorgia or something else.
Zoom in a half zoom there. Dan.
Answering that question for chat, are the black corals that you come across on your dives the same that make up some jewelry? Yes, yes it is. They are now legal to harvest, but they are the same sort of coral.
Yeah, okay, this is Victorgorgia. It's just a really nice looking, healthy colony. The white base gives it away. If it was a plexaurid, which sometimes plexaurids can be dark purple like this, the base would be darker, probably brown, but this is helpful. Victorgorgia. It's probably the similar species or the same species we've been seeing. There's not crazy diversity of Victorgorgia on here, but there are a few species. But this has been widely sampled.
DSC of that one, did you?
DSC.
Oh yeah,
working
sorry.
on it.
I got it.
Some of the branch tips at the base are denuded of polyps.
Yeah. Sure.
So
Yeah,
that
it's good.
Could have been grazing.
Ah, sorry.
No worries.
But I don't see any predators on this one either. Those brittle stars are commensals. They're probably to. There's some hypothesis that suggest that brittle stars and how they move around the colony, which is very slowly, might help keep corals free from debris and parasites and things.
That's a great close-up. I love how you can see the polyps extended in the upper right and then closed up really in the center.
Yep.
These are great view of the animals themselves.
Victorgorgia.
To answer the question in the chat, are at the summit of the seamount? Not quite. We're still making our way up.
All right, all set here in the back.
Pull wide, please.
I have a question for the ROV pilots. Does the tether connecting the two vehicles, Atlanta and Hercules, do they ever get snagged?
Yeah.
So then what do you do in that situation?
Panic, usually. Complete panic. Depends on what they get snagged on. We pick up ropes and nets are our biggest fear. Any kind of line in the water. So we carry, there's at least 3 knives on the vehicle.
Ah. Is that a new addition or has that always been?
No, that's an old thing. Most ROVs carry at least 2 knives.
Can we zoom back into this piece on the bottom?
Yeah, go ahead, Dan.
If we have a second. I'm just curious what those white
yeah, little growy things are.
Yeah. This is a sponge, probably, it looks like an old, very, very old Tretopleura.
You want to fall?
Which we've seen live specimens of on this site, but looks like there's barnacles on the fringes.
Can zoom in a bit
Maybe
more.
some other sponge colonists.
And is that manganese crust on it, or that we're seeing that black stuff, or is that just decay?
Yeah, it's probably manganese crust.
Yep, sorry.
We've collected a number of these so far.
Are you all the way in there?
Yeah, spot lobster. Yeah.
That bright white thing, is that another sponge?
That's probably a different sponge. Yeah,
growing on a
different
sponge?
species. Yep.
It looks almost clear.
Yeah,
cool.
that's really cool.
It's like a nursing log.
Looks like a mini Waltera, maybe?
There's a bunch of associates on that. Yeah, it could, maybe a polyopogon.
Yeah.
Very small one. Okay, thanks.
So, is that DSC locked up or what? What's going on with that thing? It's like an old image. Oh no, that's a current image. Sorry. I'm tripping out, man.
I'm freaking out.
Go forward.
So these kind of brittle stars that are wrapped around the sponges, wrapped around the coral, do they actually hold any benefit? Says someone in the chat wants to know.
Yeah, it's probably some sort of commensal relationship, so that means that there may not be there may be a mild benefit to the brittle star and maybe a mild or insignificant benefit to the coral.
Atlanta heading is 330.
Yeah, sorry, I lost the plot there. Still in the flashback zone.
So Dan, question for you. What is it like to control the video cameras? Can you want to talk a little bit about your role?
Yeah, absolutely. So,
the video engineer essentially works in tandem with the pilots here, and you could say we are their eyes in a sense. Not a lot of people, I think, would wager to guess that the pan and tilt are actually still controlled by the pilots, but the focus, zoom, iris, and some other miscellaneous things like color correction and, of course, controlling which feeds go to which monitors are all in our hands here. So it's a
good bar move.
Really essential position, and presents a whole host of challenges. Sometimes it's really simple when you come upon corals who are grounded and we're grounded, and really easy to snap zoom and catch them. But other times when you have
a couple meters for
coming up,
mid-water creatures, it can be really difficult to hang on and get the right shots, obviously, for both the pilots but the scientists as well. So it's a lot of moving parts, to be sure.
So what do you think is the most challenging part of being a video engineer?
That's a great question. I would say nailing focus 100% of the time.
Very important.
Yeah.
Is that a mushroom class?
Zooming and focusing at the same time, it can be done.
It's essentially a three separate things for someone with two hands. So it's just
and the iris.
it's a little ballet that we do here, but it's fun. It's always a challenge too with different animals and
okay, you can get
different things here. For example, this would be one of the more simple things to do.
Nice. Anthomastus tahinotis, a species
Beautiful.
that is I don't know if I would say endemic, but it's characteristic of the central Pacific seamounts all the way down to maybe Samoa, latitudes that far south. So it's a characteristic of the central Pacific.
That's interesting. Do we find it all over Prim? All over the monument?
All over the monument, yeah. Nautilus actually collected one at Johnstone Atoll in around 1,500 meters, something like that. So, similar depth to where we're at now. So, it's been collected.
Okay, thanks.
So, from top to bottom of the seamount, do we know how tall it is?
Lynette, can you help us with that?
I'd assume, yeah, Lynette.
Ooh, that I don't know. I know that on the dive that we're doing, we have a change in elevation of about 800 meters.
I can look
Dr.
this up.
Seuss
I was like,
sponge.
Dr.
I
Seuss
downloaded
sponge.
the
Yeah, we're going to get
Pacific
his
chemistry.
presenting his osculum here nicely. We'll get a
It
good
looks
shot.
like a loofah almost.
The yellow color is surprising. I haven't seen that.
Yeah, we've seen quite a few yellow ones. Not on this expedition, but previous.
I was thinking, oh gosh, I have not been watching very closely.
You and me both.
Is this full of snow?
There will be a quiz.
Wow.
Get off.
As they say, is an incredibly rising inflection. Shouts.
So, Steve, what are these little red, almost streaks in? Are those all brittle stars? Are those eggs? What are those?
Yeah, those are brittle stars.
Wow.
Yep. This is probably a Bolosoma or something in the subfamily Bolosominae.
Trying to balance the DSC view and the Zeus view at the
I got a
same
good
time.
pick on DSC.
It is a very,
Did a pretty
very,
good job.
very
Yeah, zoom in there for me, Dan.
speciose group,
Full
subfamily.
zoom.
Full zoom.
A lot of diversity, not well described.
Getting close.
Future career for anyone watching
yeah.
on the other side.
Sponge. I know,
Nice.
for serious,
Very
like,
nice.
please, someone. We need sponge biologists who work in the deep
maps,
sea. We
anyone?
need them.
You know, when I grow
If I
up,
could pay
I
you,
want to
I
be a
would.
spongeologist. That's
Spongeologists.
They're
That's- yep,
incredible. I mean, look at this. This is like the apartment, the condo of the deep sea, the apartment building, all the way through.
Can't promise the pay is good, but you'll never have to work a day in your life if you enjoy it.
Very true.
Wow.
Great office.
Great office, yeah. Surrounded by natural history.
I love these just
Ooh,
looking
look
at
at
like
that,
how
shrimp.
effective,
Oh,
shrimp
how
carrying
much
eggs.
surface area there
Yeah.
is.
Steer
Carrying
eggs.
eggs? Oh yeah, it
Yeah.
is too.
Yeah.
Probably a mating pair.
Mama and a papa.
Or
Love
maybe
in the deep
two,
sea.
that might be two females because they both look like they have eggs.
Oh, they
yeah.
are, yeah. Two longs.
Very nice.
So when shrimp lay eggs, do they kind of act like birds and they stick around with them, or do they kind of lay them and then leave?
Well, you can see them right on their body there.
Yeah, they will brood. Yep.
Yeah, that
Like
kind
a
of
chicken.
darker clump underneath. You stick them to their abdomen. Or maybe abdomen is the wrong body part.
Thorax.
Tail.
That's actually a good question. Would shrimp be considered insects, or it's
No.
it's just shrimp?
No, they're a different group.
Aren't they?
Totally.
Okay.
There are no marine insects.
I could spend all day looking around in it.
Yeah, that was cool.
Amazing.
Okay, which way were we going? That way, southwest.
So Lynette, how do you train to be a navigator?
It's an interesting rock that the sponge is
Oh.
on there too.
That's a good question. So I went to school for ocean mapping. So I started as doing some mapping on the vessel, and they're having their mappers cross-train as navigators, which is exciting. So we get to use the maps that we made to help navigate the vessel, see where we're going. But it's a lot of learning on the job, talking to the pilots, talking to the bridge, taking a look at all the various sensors that we have to try to build like a spatial map in your head of where we are and where we're going. Yeah.
So if someone wanted to be a navigator in the future, like what kind of skills, what advice would you give to someone to work on building?
Ooh, let's see. Communication skills for sure. We do a lot of communicating between a lot of people on the ship. Sort of spatial skills and interpreting data quickly on the fly. Like I said, we have to be paying attention to a lot of sensors all at the same time and building a visual map. So, yeah.
Awesome.
So about
also, we never did our introductions, so don't think you're off the hook, watch. We'll just do them in the second hour.
We'll do it after dinner.
Sure.
I'm Brandon, I'm from Vegas, I'm a geologist. How's that?
Brandon, how did you decide you wanted to be a geologist? What fascinated you?
The history, actually, rather than the physical specimen.
Really?
Yeah.
Tell us more.
Not human history, but just the fact that geology encompasses Earth's history was very interesting to me, and that's how I started.
What are some of the oldest rocks that you've seen?
That I've seen. I talked about it a couple of dives ago, but there's this pre-Cambrian, which pre-500 million years ago.
Oh yeah, that's right. Yeah.
This pre-Cambrian gneiss that I have, that's just outside Las Vegas. Anyone could go see it.
Can you make that full screen?
Yep.
I'm surprised I can't see it behind this thing.
Do you have a favorite
Thanks.
class you took along the way?
Yeah, paleoclimatology, which isn't marine geology, but it kind of is what got me into geochronology, which is what I do now.
All right, tell us more. For the kids at home, what does paleoclimatology mean?
Yeah, so it's studying past climates through various means, whether that be speleothems, stalactites, stalagmites, which we might see caves later on in this expedition, and using isotopes to understand past climates, using oxygen isotopes. And it was just really interesting to think about history like that, the history of climate. And geochronology is sort of a farther back version of that.
Look at this thing right there, the unbranched stalk.
Sure.
Snap zoom might be enough to do a quick confirm.
Yeah, go ahead, Dan. Have some there. Cheers.
The one up.
Huh?
It's up on top of the lasers.
Ups and the up.
Yeah, right here.
All right.
So to answer that question in the chat, aren't sea skaters a sea insect, or do they only live in fresh water? So sea skaters, from my little Google search, they from the water strider as a species, it's a subsection. They don't actually live in the sea. They just live on the sea in shallower areas.
Yeah, I'll clarify that by saying deep sea, referring to the insects. Although, you know, I think this is like the phylogenetics of relationships among the arthropods is very contentious, let's say. So everyone has their hypothesis that they support. So it's kind of like the question of which came first, the ctenophores or the cnidarians. People get very animated about that.
It's a rumble. So Steve, why is there this tiny spiral inside this bigger spiral? Like, could that be predation? Something?
Sometimes they have
strong current break off?
Sometimes their helix, it does a little reversal or double helix.
Okay.
You know, maneuver. I'm not sure. It could be new growth too.
Is it possible to have one growing inside the other?
I don't think so. That would be pretty unusual. Yeah. Yeah, sometimes if they, I don't know, there's a parasite or something blocks its regular growth, it might, adapt and create a different, little aberration in the morphology.
How you doing over there, Kukui?
I'm good. How are you guys?
Great. Do you want to tell us a little bit about your role as a data logger?
Oh, sure. So I'm a data logger, and I get to see the video feed and try to record everything that we're seeing on the screen, and
eventually when we're going uphill.
There's something over here that's going to be of interest.
Yeah, and so I also just listen to all of these professional scientists on board who also helps me with identifying all the geological and biological features that we're seeing. And I also help to take care and process the samples after we come up from the dive.
The red one, Steve?
The long stalk here? One? Can we look at the very end of it?
Right.
In detail.
In detail,
yeah, kind of right here, specifically at the branch point.
Right.
This looks a little like a broom or something.
Yeah.
I don't know.
Yeah.
Not quite trident.
It's one of those bamboo coral things. They do weird things.
Zoom in there for us a bit.
Oh, this is cool.
Zoom in a little more.
Beautiful.
That's good, thanks.
Are these some of the parasitic anemones wrapped around right at the branch?
Yeah, it could. That's what it could be. That could be those anemones are causing some sort of unusual growth. This is really useful because there are folks that are studying why certain corals branch where they branch, especially in the bamboos. So this one looks like it's probably maybe an internodal brancher. But, maybe they branch at unusual places because there's some sort of external, forcing like those parasites. This is really useful though, because sometimes you'll see them, they branch very strangely, and then, maybe the parasite goes away and you think, well, maybe that coral just branches that way.
Mm-hmm.
So having good imagery of this is really valuable.
So when the parasite takes over the entire coral, eventually, what would it look like?
Yeah, I think we're all set. Thanks, Dan.
Yeah, we saw one of those the other day that was like a different color than what we expected, and it had like smaller polyps than the skeleton that it was on. I thought that was super cool. We saw like the half-half one, and then the full one. I like the half-half ones a little better.
Good, go white, thanks.
But I guess do these anemones
sure.
ever go bonkers? Do we get many anemones on a coral, or is it usually just a couple?
A couple, one or a couple, yeah.
Okay.
Yeah. Sometimes what can happen is if the coral is able to, it'll form a gall and kind of surround the anemone, and it'll encase it in calcium carbonate and suffocate it that way, such that it kills it, and it isn't a threat to the coral, because
We're just
the worst thing for a coral is to have exposed skeleton that can be left open to
quite a bit.
fouling organisms like hydroids or other animals that might wish to take over your colony.
Southwest.
Which bearing are we moving?
Southwest.
Okay.
Quite a lot of rock debris
definitely
here.
You got a
Must spin around.
sonar action there.
Yeah,
Reminds me of scree slopes.
we're starting to climb again up a very gentle slope. Doesn't look very gentle, but at least that's what it suggests on the bathymetry.
So Kukui, do you feel like you had any classes, or I guess same question I threw to Lynette, like what kind of skills or what kind of classes would you advise students to take if they're interested in following a path like yours?
And do you have any advice for kids trying to get into that field as well?
Oh, yeah. Well, I'm kind of a student who's all over the place. I took a bunch of different classes as a marine science major at my school. Biodiversity was a huge help, as well as oceanography, physical, chemical oceanography, geological oceanography. And also I've been trying to find classes that will help me integrate more of a indigenous culture aspect into science. So that's why I went into a Hawaiian Studies minor. And so I've been really blessed to be able to take a lot of Olelo Hawaii classes, as well as Ike Ku'una, or like traditional knowledge classes. And for advice, I would just say just full send, apply to everything that interests you, apply to everything that you can, and yeah, just go for it.
I think that's such good advice, and sometimes hard to take, you know, of kind of like, do you have any strategies for how, you know, you continued putting yourself in those opportunities for those paths?
I just continued networking with as many people as I could throughout all the programs that I was blessed to be able to take part in, and trying to get to connect with them on a personal and professional level really helps a lot to be more comfortable in networking with more and more people.
So for everybody at home, where are you in your degree path, and what's coming up next?
That is a very good question. I have one more semester left, hopefully, for my undergraduate, and then I have no idea.
And you know what? That's okay.
Thank you, Matt.
That's how I was after my undergrad too.
Same.
Oh, yeah, it's very comforting.
Same.
The
Very comforting.
world is your oyster.
The world is your crinoid.
And what about you, Megan? What kind of skills and classes and pathways did you take to get here?
Also very windy. I'm trained as a biologist,
Umbellapathes.
and doing science communication is something I've moved my career into and built, and program management after that. I think science communication starts with a lot of passion and respect, like both of those things, that you want to help share the things that you're really excited about, and also you want to connect with people and share them in ways that they can kind of like dock up to your excitement, you know, whether that's connection through
this
like
crack right there was something shiny.
Ooh,
right
shiny.
science or ocean or whatever. yeah, I think networking is really big. I also, there are so many skills in the umbrella of science, and
Little coral on the rock there too.
I'm really passionate that science
Go
is a
ahead and
team
zoom in there a
sport.
little bit, Dan.
Yeah.
And that it takes a whole lot of people with a lot of different skills to make it successful, and that's a thing I love about Nautilus. So I've always felt like I'm a really strong writer, and so that's helped me, translate my ideas from just like ideas.
Not seeing it.
Gastropod.
It's that thing there. No, it's just it's a base of a colony. Old base with some deep sea mucus. I thought it was shiny. Okay.
A little cup coral right there. Look at her, so cute.
Tiny cup coral.
It's awesome.
Oh, is that like a singular polyp?
We're good. Full zoom there, Dan.
Yeah, you bet.
Baby coral.
Yeah.
Oh,
Yeah.
Full, maybe full size?
Actually,
That's awesome.
have we any slurp jars?
Yes, we have, I believe, 4 slurp jars available.
Oh yeah, let's slurp this.
What's that?
Can we slurp the cup coral?
You want to slurp that cute little baby cup coral? Roger that.
There is
Dan gets
a
really attached to the
scientific
cup corals.
objective for this.
Clearly.
I'm over it. You have to go wait for me to get the toy out here.
Yeah, there is a scientific purpose for this.
Am I good to start flushing?
Sure.
But yeah, I think you can like learn a lot of skills, and so I think advice in general for people who want to get into really whether it's science communication or expeditions or whatever, it's just like be willing to listen and take feedback, be willing to keep learning.
It ran away.
That like
we can still use it.
Yeah. Okay, zoom back out.
There's no one in the world who is going to be
easier to get it off the rock if you kill it first. With the
Thanks for
ROV.
that. We'll pick it up.
We're really interested in the tissue, so
go to some.
We'll make some lemonade out of these lemons.
I'm sorry, little one.
Lemonade.
Looks like there's something in that one. Okay, can zoom in, Dan.
Number 4 should be empty, yeah?
Correct.
Can't believe we hit him.
So this is a caryophyllid cup coral.
What do we know about caryophyllates?
Good. Okay.
It is a family of solitary scleractinians that is very common in the deep sea, but this group is being investigated by some of our scientists ashore who have submitted requests for any caryophyllates we come across. USGS, the U.S. Geological Survey. Yes, they do biology too. So they're interested in population genetics.
Not gonna tell us how we're gonna get if we got it in or not.
Caryophyllid cup corals, and so this will help.
You'll see it in there.
Accomplish. This will help help our scientists ashore.
Answering that question in the chat. Do you have a dedicated IT person aboard? I believe we do, right?
Absolutely, we do.
Absolutely, yes.
Okay, you can turn it off there. See if it came through.
Tim Burbank is our data engineer. We call that department data engineering and data systems.
Tim's got some cool hair too.
Yeah, he has a huge job. Every
turn it back on.
Instrument here, camera, computer that is generating data about this expedition. He has expertise in all those systems to help them come together, to help them be archived, to troubleshoot along with all of our technical teams here, keep the satellite up so that we can live broadcast and stream.
Okay,
there it is.
If you want to learn more about that, check out the team page.
That is sample 060.
On the team page, there are dedicated videos and write-ups about every job on board, and some of the people who serve in those jobs, what a day is like in the life of those folks.
There's some disgusting stuff in that jar.
Advice, what you might want to get him, do if you want to go on a path like that. That's a huge field. Ocean data is just like an exploding field as we advance technology and really try and catch up to the work that we have to do, which is understanding the majority of the planet, which happens to be salty, and all of the impacts that we're having on it. There's just going to be more and more and more exponential data. And
oh, you want to change that camera back?
Combining computer science and a passion for the ocean is a huge path. Lots of ways you can get involved there.
And it's really cool to look at some of the stuff that has never been seen before by human eyes.
I got a 6.8 wrap. Do you want to take that out now?
Yeah, maybe cooler than working at like a giant software company, right? You could take the same skills and you could apply it to a different setting.
Exactly, and you could be on a cool ship.
But yeah, so I definitely check out for any students listening, anyone interested in
where's Herc, Mike?
Herc is right below.
Interested in those career paths or trying to follow them, check out everybody's bio. You can click into them just below the live feed. And we've all filled out some questions about other jobs we've had, advice we'd give to someone who wants a career like ours. Great resource.
And I'll say, Megan, too, those bios might actually land you a job on the ship, too, because that's exactly what happened to me.
You think we could grab a rock?
Tell me more.
Yeah. I think we could grab a rock.
That was
would it look useful to you? What do we got?
Circle one that you like.
I believe we
should. Let me start looking.
Just like the QCed network.
This guy right here, actually.
Yeah,
sorry.
You probably have some room, too.
Totally going to come back to that story.
All right,
we're not
good. Plenty of room.
You're not done, Dan.
Ready to go. This guy.
Pick up that rock, Mike.
Take out the 6.8 wrap?
No, never mind.
I can't
I'm going to
hear
be
you.
quick about it because we're on the move.
No. Circle it again.
Yeah.
All right, Brandon, what did you spot in this rock that made you want it, this one?
It looks like a good size, and it looks somewhat angular, I think.
This is Brandon's rock. There are many like it,
and
but
it's
this
thick.
one's his.
Not too thin.
Happy with that one?
Yeah, that looks good.
Okay, Mike, I'm going to open the box first.
Open stopper box.
Awesome.
What's that? Before you do that, hit 5 on the bubble cam for me.
Is it, did you want the toolbox or you want the starboard box?
Starboard box. Starboard box, some box that I don't know yet.
D?
I'm assuming you got an image of it there.
Yeah, we got it.
Nice.
Delta.
Let me know when you're ready for salvo.
Perfect.
I don't need a salvo.
Okay.
What box?
Delta.
Delta. Which one's Delta? A, B, C.
All the way in the back.
D. No, the rock ain't gonna fit in Delta.
Not in Delta?
Alright, gonna try it.
Uh-oh. If you're watching on channel 3.
Nice size.
Like a glove.
Totally.
That was awesome.
That's funny, me being pessimistic. Usually I'm fatally optimistic with what rock will fit in the box.
That's not gonna fly.
That'll never happen.
Alright, closing box.
Roger.
Last year there was a rock on, in the starboard bio box that left about a 1-inch gap.
Definitely seen that where everyone holds their breath as the tool tray comes back in, hoping it'll close.
Hit one on bubble for me.
I mean, it's not bad. It's just heavy. I think the heaviest rock that I've ever sampled, well, not me, but I've ever had to carry,
Dwayne Johnson.
that has been sampled, was around 90 pounds last year. It's heavy. It's not a rock anymore. That's a boulder.
Totally.
Boulder can. That's great.
It had its own box. It was so big.
We had some of those during our latest expedition, 138.
Sorry, I'll try and DSC it.
Here's another question for chat. I'm 45. Is it too late to get involved with work on the Nautilus?
It is not. There's no age range on our internships. If you are in school or an early career professional, no matter what age that is at, if you're interested in working with us as a contractor, you can check out the join page.
Polyophagon sponge.
Each department this late summer, fall will open up our contract opportunities for next year, and that's the way we invite people to get express their interest and let us know if they'd like to join us for the season ahead.
Good shot. Yeah.
We do kind of bulk hiring. Same thing, our science and engineering internship program and our science communication fellowship applications will all open up this fall. And we'll staff the entire 2023 season that way.
Cool. So going back to getting jobs, stuff like that, Dan, I believe you were telling a story. Not Delta Dan, Cameraman Dan.
Cameraman Dan. Yeah, no, not so much a story, just as a little bit of advice to any students, whether high school undergrad or even graduate. Just scouring those crew member profiles, those bios that are on Nautilus Live. If you have any interest whatsoever, and this kind of plays out the question that just came into chat. Age is really not a factor, and there's a multitude of disciplines, different skills that are utilized here, from video to the ROV pilots to communication, data logging, etc. There's tons of different skill sets you can get into, and I found it really invaluable years ago when I was first interested in deep sea research by just reading. I read over dozens and dozens, and managed to send out a couple emails to the Ocean Exploration Trust and get on a few Zoom calls and talk with a few people, and years down the line it led towards me getting a call, towards getting on the ship. So this is my first cruise with Nautilus, and I've known several video engineers in the past who been fortunate enough to come aboard, and it really is a fantastic experience.
Look left a little.
I would say don't let any
Michael.
It doesn't necessarily matter too much what your background is. Mine is pretty similar to Megan's, actually. I was a biologist and kind of fell into television and filmmaking by the wayside, and took an interest in broadcast as well, and those skill sets just so happen to really suit the position of a video engineer. So it really is Nautilus is really available to people with a very broad range of skill sets, and I think so long as you have passion for deep sea research and exploration in that regard, reach out to people, email them, talk to us. So many people here are more than eager and willing to help and give you advice and give you a hand.
Yeah, I think you totally nailed another theme, which is persistence. Persistence is really important. The Nautilus programs are competitive. There are fewer than 20 people who work for Ocean Exploration Trust full time. There are maybe about 150 who come to sea in a season. I've never ever knocked on somebody's front door and said hey, do you want to come to sea with us? We select people from people who express interest and put themselves out there. But sometimes that may be, a couple seasons, or it may be gaining different experiences and bringing them, kind of triangulating in on your career of choice. I had a lot of different jobs before I decided what I wanted to do, and what I wanted to do has changed throughout my career, year by year, or kind of season by season. So just continuing to search for ways to get to your goals is so key. And a no is not so bad, right? A no is just an opportunity to say yes later.
It's true. As first someone says no, just keep on trying, and eventually they'll say yes.
Yeah, ask for feedback, ask how you can keep learning and growing.
Yeah.
And full send.
And full
send. Full send.
I love it.
I like that, Kukui.
Thank you. That's a beautiful, I think, Polyophagon species.
This one is so dense, it just looks like it's like a shoebox of a sponge.
Looks almost like a potato in the kind of consistency.
It's like jutting into the bumper.
Sea
We're approaching dinner. We're like,
It's
it's bread, it's potato, it's
all the analogies will be food.
yeah, exactly. Nice.
Oh man, now I'm getting hungry. Oh no.
So if you are just joining us, you are looking at an extremely zoomed in section of a coral, or sorry, of a sponge. We're taking a good look at the spicules there. Polyopicon sponge, I believe, in that group. If you're watching on channel 2, you're seeing the Atalanta view looking down at the seafloor at Hercules. And if you're watching on channel 3, this is the control room where we are all sitting here aboard the EP Nautilus with the vehicles down below.
So that on that zoom, you could even see what gives the hexactinellid sponges their group name, which are a specific type of the sponge spicule that look like these little umbrellas. We were zoomed in so close that you could actually see the little umbrella shapes on the top there.
Beautiful.
It was really cool.
And I believe that was the Stauropathes black coral we saw in the background of the Polyopicon. Nice.
Or as we like to call it, a sponge critter.
All right, Kevin, you're here. Welcome for dinner relief.
Hey.
Hey. What? Tell me about these rocks.
They're all fantastic and beautiful and perfect.
Tell me more.
No.
We've been traversing a seamount that was never an island, so it's always submarine, but it has these steep, beautiful basaltic flows. We got a range of types. These are more pillow, low bait style flows right here. We've seen sheet flows, we've seen dikes, we've seen columnar jointing. We've seen a little bit of everything so far this dive. And
Yeah.
we
Let me bump up a little bit.
We started kind of on a mass wasting deposit, so a large slope, basically a big mound that was just full of chunks of basalt. And then we went up a steep cliff where those chunks of basalt fell off of. So we get to see both ends. And I think we've gotten like 8 basalts so far, so I'm very happy. I just cut up all the ones from the last dive. There was 1 or 2 good, decent looking basalts. The rest were all mostly ferromanganese crusts that gets like centimeters, like 10 plus centimeters thick in this area. So really thick ferromanganese crusts. And phosphorite, which is a calcium fluoroapatite, where limestone turns into. So these kind of coral limestones can change compositions through time into something called phosphorite. So we found a lot of that. Definitely confirms that we're dealing with really old seamount, greater than 90 million years old in this region.
Do you generally find the mass wasting events on older seamounts? Is that an indicator of age, or is it an indicator of something else in its history?
No, it's common on almost all seamounts to a certain extent. And most of the time, any kind of large mass wasting event occurs pretty close to when it first formed. It's usually when it starts to subside because the heat source is away and it's no longer active. It will start to sink into the mantle and get denser. And during that quick early period when it first starts sinking is when it can kind of fault and fall apart. But you can have very large sector collapses. Like north of Oahu is like a quarter of the island basically came off about 5 million years ago, and you can see it on Google Maps. It's quite dramatic. So it's not uncommon for these seamounts to have large, collapses.
Is it also common to have such a variety of flow types within a seamount?
Yeah. Yeah, flow types usually is a function of 2 things. Flow rate is the primary thing since we're mostly dealing with basalts. So when you have really low flow rate, where it's just like squeezing a little bit of magma out of a time, it will kind of cool into those pillows, and you'll get distinct little round pillows that kind of just fall out of the vent. When it gets a little faster, you get those low bait flows, so it's kind of pillow, but they're elongated, where you kind of quench the top of it, and then slowly fill it with lava from the inside, so it expands outwards. And then if it's really high flow rate, you get what are called sheet flows, which are thin layers that kind of cool and keep moving and cooling, and you get kind of stacks of thin flows on top of each other. Now, when you have higher silica contents, so then they get more evolved and you have longer-lived magma chambers, you can get higher viscosity, and with higher viscosity, you can get thicker flows that don't move as far. But yeah, you can find all of the above on the seamount.
As we unfold the history of the seamount, will we know if it was kind of continuously erupting, or whether it was a variety of, in 100 million years, you could have a lot of eruptions separated by a million years and be building this thing over time?
Yeah.
Do we need to be sampling a lot more to tell that?
Typically, yeah, you want a lot of samples. From what we know of seamounts, they usually last about 5 million years. So it takes about 5 million years to construct it, and they stop erupting after that. What can happen, though, is you can have reactivation. So you can have a seamount that formed from, say, mantle plume or some other mantle anomaly, and then move over another mantle plume, say, 70 million years later or something like that. And that's actually what we see in the Johnson Atoll in some areas, where it appears like we have 100-million-year-old volcanoes that then get overprinted by 70-million-year-old volcanism. So there's multiple mantle plumes. The lithosphere of this area has gone over multiple mantle plumes in its 115-million-year history.
Amazing, thank you.
Yeah, I'm going to hand it off to Brandon. He's doing a great job.
Welcome back, Brandon.
So wherever you are in the world, we're thrilled to have you here with us. It is 5:30 p.m. Hawaii time, which means we're right in the middle of dinner and a dinner swap out and handover. So we end up with kind of like a really fun mix and match watch team as different folks come in to grab food.
It is a little bit of a wild card hour.
Mm-hmm.
Well, it's usually our same swap out team every night, but you get a slightly different combination of times.
Exactly. Who's going to come when? When will we get to see Meredith? It's like saved rounds. It's like every eDNA question. Wait.
Well, I'm happy to answer any of them, so.
All right, team, who has an eDNA question for Meredith?
Why is eDNA important to collect?
eDNA is an important component of this type of visual survey. You can also use it standalone from a blind survey like a CTD cast, but the nice thing here is we can pair this visual imagery with the eDNA sample and confirm what species we're looking at. But then it also gives us a bigger picture of this environment. So because this is a visual survey and we can only see what the lights from the ROV and the cameras are seeing, we could be missing things that are just outside the light pool. We'd have no idea, no way of knowing they're there. But if there's enough of them around, they have an eDNA signal that we can pick up. So it gives us a slightly different but complementary view of the ecosystem.
And with these eDNA samples, how do you sample them? Do you go through a machine? Do you test the water? How do you test them?
So the first step is taking a Niskin sample with the ROV. We've already taken 4 of those so far this evening. So we have the pilots just trigger these bottles. They have lids on both ends that snap closed and capture water from that area, and then it stays captured till we get the ROV back to the surface. Here on the ship, I filter that water down, and I take the little filter membrane and I preserve that in a DNA buffer. And then that goes back to the lab in Seattle at the Northwest Fisheries Science Center, where I will be extracting DNA and then sequencing it for octocorals.
Oh. So are you only specifically looking for octocorals or other things as well?
In these samples, we're going to look for octocorals. We're also going to run them for the fish community. We'll probably have some mixed success with our ability to amplify fish just because there have not been that many in the area, but we're going to try. And then we may be working on developing some new markers to run associates, which are the little invertebrates we're seeing in the corals. But we'll see how far we get with these samples with octocorals first. These metallogorgia corals have been really abundant throughout the stat.
Starting gauge checks, Chris.
Looks like there's some associate. Is it a lobster? Squat lobster?
I think it's a squat lobster. They've seen a couple of those up on this upper slope, just kind of hanging like little Christmas ornaments in the corals.
Do you want tighter, Chris?
Yeah, a little tighter.
Yep, nice little squat lobster there.
Okay, come wide.
Coming wide.
Is there a massive cucumber over there on the right side of the screen?
I do believe there was. Big kind of pale lavender color up in the upper right-hand corner. And what we're looking at right here is a little umbellapathes, or possibly alternatipathes black coral. We've sampled one of these on the last watch. This is a group that's currently undergoing a little bit of taxonomic revision. So one of our scientists ashore asked for an additional sample.
All right, come wide.
Coming wide.
Coral skeleton there.
No, I didn't.
Although actually, no, looking at it through the still cam view, I think that's just it's a very tall black coral.
Half zoom.
Half zoom.
This is probably Parentipathes black coral.
With a crinoid on it?
It's a crinoid on it, and it looks like it has another little squat lobster as well.
All right, come wide.
Full wide.
Looks like a sediment field over there. Maybe nodules, perhaps?
So far, everything we've tested to be nodules has not turned out to, in fact, be nodules.
No
it's
nodules.
all just been basalt rubble.
Yep. Wow, this is quite flat. It's a gorgeous looking thin sheet flow right there.
All right, zoom.
What's the name of this sponge again?
Okay, full wide.
Full wide.
Walteria?
Walteria.
Walteria. I knew it was similar to the name Walter. So.
Atalanta and Hercules gauges look good.
The sponges have been all over the ridgeline we've been following up. They've been the most common sponge we've seen all day.
Yep.
It's interesting how right on the top of this ridgeline, it's so
It's going to come up a
flat.
bit. I'm just watching the sonar
Right.
here.
And then the minute you get right to the edge, it just drops off in a huge, huge cliff faces.
Yeah, we were theorizing that perhaps there was some kind of landslide feature on the seamount.
Has there been any evidence of that so far, or are we still gonna take a look at another waypoint?
Yeah, I think there has been. Pretty much along this ridge we've been following, we've seen these debris fields where the outer, like the side walls are lifted.
Oh, that one has a really nice tight spiral there.
All right, can we get a zoom video?
I think this is going to be possibly a Rodana-Ridgegorgia. With that really tight spring coil stalk.
It's a thick sea star under it.
Mm-hmm. That, yeah, I think that's the sea star that may be predating on this coral.
Oh, interesting. Yeah, it doesn't look like a brittle star.
Nope, that is a full-on.
All right, can we
star.
get a full zoom?
Full zoom.
Oh yeah, you can see the polyps that are missing from the branches right where it's sitting.
Oh, yeah.
Such a nice view of it there.
Predation in slow motion.
Got a little bit more zoom if you want me to get.
Yeah, go on in,
okay.
and we'll move on. Nice dark backdrop. Okay. All right. Can you come a little wider?
A little wider.
There we go. Oh. I'm going to come up for a second, watching the tether there. Oh. There's no wraps in it. It's just looped around itself there. Yeah, I'll hold here. We'll let Chris pass underneath me and should straighten it out.
I wasn't on SPL. We're trying to get a shot of his tummy.
Ooh, that's a good still.
Yeah, I got some good stills here.
It's amazing how much Atalanta kept drifting after the vessel stopped there.
That is.
Oh, there's another sea star on the base. I didn't see that one.
Yeah, I didn't see the one on the base either. Calling his friends.
So these corals are not ideal for this predation sampling we're interested in, mostly because there's not enough massive tissue really in one of these colonies to really make it worth our while.
Oh, you want to sample one?
No, yeah, but we're interested in sampling a different family of corals that's being predated upon. So not quite this one.
But you want to sample the sea star that's predating?
No, neither in this case.
Just, no, not in this case, but
yeah.
what you're looking for is.
Yeah, yeah.
So you want to see what the sea star is?
Not particularly in this case. I know it's predating on the coral. You can see a bunch of stripped branches.
Yeah.
Yep.
That's a nice shot.
That's the money shot.
Heck yeah. Whoa.
What happened to our bubble here?
I don't know. Hit one, enter.
Oh, there we go.
Cool.
Okay, come wide.
All right.
Nice.
Nicely done, Chris.
Thank you.
If you catch up to Atalanta there and mind the tether while you...
Yeah, tether's looking better now.
Yeah. Turns are all good, so...
Yep. Paper here with some drawings on it.
If he comes under you, Gibson, you can come up, but if he goes to the side, you can like come halfway up.
Might have taken it already.
Roger that.
There are many things.
Either way works.
Yes, that one. No. In total, we need about 500 milligrams of tissue, which this much of Chrysogorgia would not satisfy. So primnoids are ideal. Paragorgia is pretty okay too.
So it's the coral that you want, or the?
It's a specific type of coral undergoing predation, usually we find these sea stars on the primnoids or bamboo corals almost always on every dive, but it's been very rare the past several sites.
Can I get a DVL reset, Dan?
Sure.
Sometimes you come upon a site and there's dozens of predation instances to choose from.
Reset DVL. Yeah, it'll just set it to the USBL. You don't need to click. The USBL has been good enough that you don't really need to.
All right. Okay, the professional's back. I'm out.
Yeah, I could do another 15 minutes or something if you... I could do another 15 minutes or so if... Okay. All right, Dan's gone. Let's see what this thing can do.
Turn it up to 11.
Question for the ROV guys. Why is Atlanta being used instead of Argus?
So I think one reason we're going with Atlanta is because these dives have been deeper than we're used to going, and Atlanta is significantly smaller and lighter than Argus.
You can put in another ship move.
Which takes some of the load off the winch, because we got a lot of cable out. think there were also some maintenance and component reasons to switch to Atalanta. There's some stuff in the shop at the moment. Sounds like working with Atlanta has been a bit of a learning curve. It swings around a lot more behind the Nautilus. A little less predictable.
Hey, Chanel.
Yeah, what's up?
Do you know what sample number that last basalt we collected was?
I believe it's 061.
Okay, thank you.
Can we look closely at the branch point on this long stalk?
Roger.
Oh, interesting. Midway branch point.
Yeah.
Is that a bamboo coral?
It is.
Might need a second
yeah, it's
here.
kind of the same as the last one we saw that was branching multiple times around where they had that anemone.
Interesting.
So I'm specifically interested in kind of like that section.
Okay.
Might feel a little tug in a moment here, Chris.
Yeah, I know. I'm already kind of feeling it. we gotta wait a second for the ship.
Yeah, sure.
We can do a partial zoom at least for now.
Looks to be the same kind of scenario. There's an anemone and then the coral is branching.
Okay, full zoom.
Full zoom.
Sometimes multiple branches at each anemone site.
There's an anemone there?
Yeah, right here.
Oh,
That's
wow.
the anemone. It's in the back, yeah.
Yep.
So it looks like it might be branching at a node point, but I'm not sure if that's because that's just where the anemone is or if it's a nodal brancher in this case. Although certain other parts of it suggest that it's branching at internodes, so it might not have a clear pattern. Same thing here, there's some sort of obstruction. It looks like it's forming a branch point just above the lasers.
Very cool.
If you want, I can kind of pan up the length of it.
Yeah, sure. Now you can see the baby branch here, just starting to bud off. It looks like it's branching at the node.
So when these corals start essentially calcifying the sea anemone, what does it look like?
When they start calcifying over the anemone?
Yeah.
If that's what this is doing, it doesn't have to be that. But sometimes they'll form multiple branches that'll form like a basket and smother the parasite. I'm not sure what this one is doing, how it's compensating. It sounds like it might be creating extra branches to maybe, I don't know, just to, avoid having yeah, I don't know. I honestly don't even know in this.
All right, we done with this?
Yep.
All right.
We're all set.
Come wide.
It's tough to say why they're doing that.
Full wide.
But there are people studying why they might branch at certain places, and so hopefully this data will provide them useful information to help us determine why they might branch in certain places, and then if it's related to animals that might be attached.
So can coral defend against those anemones in other ways aside from calcifying it?
Yeah, they possibly. Anemones are pretty successful at attaching, and sometimes the anemones can live aside with the coral without issue. We see sometimes numerous anemones in coral colonies, but sometimes they can get very big and become a problem for the colony. They don't add to the calcification process, so it represents kind of a weak link in the branch, in the colony. So they can potentially fight using kind of a chemical warfare of sorts, where they might produce unpalatable chemicals for the other animal. Anemones also have kind of mechanical defenses. Sometimes they can produce specialized tentacles that will attack other cnidarians in the area, so kind of a defense mechanism. So stinging other corals.
So with these corals specifically, when we collect them, is there a way that we can accurately age them? Says someone in the chat wanted to know.
With the bamboos, it's possible. Yeah, you would have to kill the entire organism right now using the methods we currently have available to us, which is not ideal. Number one, they're extremely large animals. So getting the whole animal, making good use of it, getting it to the surface is impossible without having to cut it up further.
Yeah.
So for the most part, we're happy just getting visual observations for now. But there are scientists in the world who are specifically studying that, and they have the ability to go out and collect those data.
So now also, when the sea anemones attach to this coral, is there like an average lifespan of the anemone on the coral?
Unclear. Yeah, unclear. Very, very difficult to tell. The only way you can really tell those things is if you, for example, have a known reference point where, like Dan was saying earlier, a cable or something was laid, or a shipwreck, and there was some animal that you had it track the time series of through its life from settlement all the way to death. You might be able to collect that data, but from the environment we're in right now, using the tools we have right now, we wouldn't be able to tell that. Can we do the same thing on this colony that we did in the last one? Zoom on those branch points?
Sure thing. Might need a little more tether.
Coming down.
As closely as possible. This one's being eaten as well, which is why all the polyps are closed up. There's a bamboo coral down at the base.
Let's do a half zoom.
Half zoom.
Okay.
This is interesting. This is it looks like a totally different species of unbranched with air quotes, bamboo coral. The polyp morphology here they're more lumpy, bumpy, whereas the other ones were more cylindrical. The polyp
all right, can we do a full zoom?
Is fairly erect. So that tells us something about what might be causing the branch
all the way in
initiation here. It's probably the anemone, since they're all kind of doing the same thing.
So you said it's being eaten. Is it being eaten by that anemone?
Can we kind of walk up to the second branch point that's just north of this? Yeah, I don't know what's going on with the anemone here. If it's if the host coral is just adapting. So this kind of looks like a internodal brancher, but it's hard to tell. It's very heavily calcified at those branch points. sometimes that can hide the
Is this the
presence of
branch you want to
node.
look at, or?
Yeah, that's right.
Okay.
All right, I think that'll be good enough.
Okay. Full wide.
That's great. Very good.
Full wide.
That is a really bizarre branching pattern, though. It's
Yeah.
not anything I've seen before.
Oops.
We have a follow-up question for that anemone thing. What happens when an anemone attaches in between the nodes of a nodal branch coral? Do they still form new branches around the anemone?
That's a great question, and that's what we're trying to find out with these data. So there was a scientist out on our earlier cruise in the season to Kingman and Palmyra who is studying just that question. Hopefully their results will help shed some light on that question.
Is there any hypothesis or theories around it?
No, it's primarily just observation right now. There's a lot of different things that can cause branch propagation. I think the main thing to find out right now is it something in the taxonomy of the coral that's causing it to branch in life, or is it some sort of external, forcing, like an anemone or a parasite or a wound or something. But some corals just branch naturally late in life, and they'll produce very sparse branches. So you could imagine only seeing snapshots of bamboo corals that are unbranched in their life. It might be difficult to have a whole picture without seeing lots and lots and lots of different corals and looking at their polyp morphology and growth patterns.
This is part where?
Bubblegum coral down there. Very small iridogorgia as well.
Can we get a quick zoom? Okay. Whoop. Okay, come wide.
Another bubblegum coral. Also some Chrysogorgia around here. So small puff balls in the rock. All things we've seen before. Looks to be a small zoanthid branch. Possibly on the backside of this colony. Oh, there's an umbellula too. Umbellula sea pen right here in the background. Oops, let me get the...
Can we do a full zoom?
Yep.
You can close those screens on the, if you pull it up on the Herc-Ross thing.
Yep, crinoid there, brittle stars.
Chris is
Looks
saying
like
you
it's
can close
missing a
those
few
down.
of its
We
appendages.
can close
Do you know
those
why that
screens.
is?
The
Yeah,
crinoid?
Yeah.
Press the Herc-Ross button on that.
Yeah, unclear. I mean, they do have grazers that feed on the arms of those crinoids. Then again, they do regrow them as well. So it's not clear if it could be in regrowth or if it's just been recently lost. Snails often will nibble on the arm tips of crinoids like that. Maybe a fish or something. All right.
All right, let's full wide.
Thank you. All set there.
Oh, it's probably taken up downstairs. You can, here, kill it there. You got to use that, keep that screen, or that station. Yeah, Herc-Ross proc. Just click the X. Yep. And then it'll disconnect it, and then you can take it. Yeah, now you can take the screen. Yeah.
I'll throw it up in pip 1.
It's actually kind of handy to tell where your back end is. Yeah.
Seeing like evidence of like sheet flow just covered by sediment and other debris. See sort of these thin rocks. Well, here you can see the sheet flow better.
Is it easy to tell how long ago a sheet flow is, depending on how much debris is on?
Long ago?
Yeah, like can you age it just by looking at it, or do you have to cut into the rock?
No, I have no way of knowing the age by looking at it.
So what are you looking for specifically?
To know if it's a sheet flow or?
Well, that too, I guess, but to age it, and I guess if it's a sheet flow.
I'm just looking for good samples to pick up to age it for later.
Oh, you got a, what is that there? Flytrap anemone?
Oh yeah,
Oh,
to the
yeah.
bottom
Very
right.
bottom, yeah.
Oh, wow.
You want to take a look?
Yeah, we can do a quick zoom in, and
right,
we don't have to
let me get
linger on
downslope
it.
a little bit of it here.
Haven't seen very many of these. Usually they're very common.
Can you think of any reason why we haven't seen them?
Half zoom.
No, I don't really know. They're pretty universal species. Found them almost in every ocean basin. Very common in lower latitudes. Atlantic, Pacific. It's not that many here. This one's attached to an old coral stalk, most likely. Some barnacles on the tips, but you can see why they're kind of called flytrap anemones. But not plants.
So what do they primarily feed on?
Invertebrates. Or anything that comes and drips into their tentacles here, but
So
mostly invertebrates.
Did he grow around the stalk like that?
All right, come wide video.
Yeah, that's what they do.
Wow.
Yep,
And so they're sessile, right?
that's right.
What does sessile mean?
They don't move when they're settled.
Okay.
So do they actively look for dead coral, or do they kill the coral, or do they just attach to whatever is available?
Good question. Probably all of the above.
Nice.
And prior to attaching to a stalk, are they just sort of wandering in search of a home?
They probably settle there as larvae, so they're not walking around, looking for a good spot. They probably settle very
Early on.
very young out of the plankton. Yeah.
Understood.
So it's not necessarily a stalk only that they settle on. It could be a rock.
Yeah, I mean any substrate that is above the, above the seafloor, a few centimeters, is even preferable to something on a rock.
So you're saying we won't see any walking fly traps?
No.
Shucks.
But if you did, it would be a pretty big deal.
I would give it
All
5
right.
stars as
I'll
a
keep my eyes
highlight.
open.
Anemones do, they do have some locomotion. They do have some ability to move. Did it make a highlight video earlier this year with the purple anemone? We captured a video in March where we tried to slurp a purple Ceryanthid anemone and it started to swim through the water column.
It did not, but I do know the clip that you were thinking of.
Yes. Everyone was very excited about that.
It might make like a sample compilation.
Yeah. Yeah, it was
so cool.
It was a local favorite here, but
You know those, I don't know if anyone who's approximately the same age as me who's watching all this live, but you know those really squishy